Alright guys, I'm going to post a kind of a walkthrough video on the chicken genetics gizmo that you guys were supposed to do a while ago. Some of us are still working on it, so I figured I'd give you some help here. I'll walk you through some of the steps or kind of what you're supposed to do um, on the gizmo so that we can get this done and out of the way. The first two questions on the gizmo are associated with this red flower here. You guys are supposed to look at it and kind of figure out what you think the dominant petal color is, white or red. And if you remember, this is a gizmo kind of helping us figure out what codominance means in an organism. Codominance being that there's multiple dominant traits and not just one, so that both traits will show up. So it's going to talk to you guys about that. Second question kind of goes off further on that, and then we get into the gizmo itself. So what I'm going to do for you guys is I'm going to flip over to the gizmo so that we can practice that. Real quick. All right. So if we look here, the first question was turn on the show genotypes. The genotype is the allele combination in an organism. And it says point to the red chicken. So we click that on, turn on full screen. Point to the red chicken, it says FR. And then we point to the white chicken, it says big F, big W, big F, big W. So that's what you'll fill in for number 1 there. Number 2 says, what do you think the letters F, R, and W stand for? Hopefully it's pretty self-explanatory when we're talking about feather colors. Big hint there. So go down to activity A and it says get the gizmo ready. It says drag a red chicken and a white chicken into the parent boxes, but don't click breed yet. So I drag the red and white chicken into the breeding boxes. The first step, number 1, says predict. What do you think the offspring of the red chicken and the white chicken will look like? Write down your prediction there. Number 2 says observe. Click breed. What are the offspring's genotypes? So as we breed them, you'll see that there's 5 chickens that pop up, and they all look a little bit different than their parents. And so you'll tap on one of them, and you'll say, okay, all 5 of them have big F, big R, big F, big W. The organism's appearance is the phenotype, and what is the phenotype? And as you're looking at each of these here, the phenotype, as you can see, is white and red feathers. Moving on to number 3, it says experiment. Drag 4 offspring into the holding cages. 1, 2, 3, 4. Click clear, and then drag 1 of the offspring into the parent box. Drag a white chicken into the other box. Click breed several times. Describe the resulting genotypes and phenotypes. So I'll drag an offspring and a white parent up there. I click breed several times. 1, 2, 3, 4, 5. Now, it's asking for genotypes. So our genotypes are, there you can see, they are FWFW or FRFW. And then the phenotypes for those would be white chickens and white and red chickens. That's what you'll be filling out in that section. Number 4 says revise and repeat. It says click clear, drag another chicken from the holding cages to the parent box, and then drag a red chicken to the other box. Click breed several times. So I'm going to clear, I'm going to do a red chicken this time with a mixed chicken. Click breed several times. Okay, and here you can see, based on the statistics, we have offspring that are red, FRFR, and we have offspring that are mixed, FRFW. that express both traits. As I go to number 5 says explain. In dominant recessive inheritance patterns, the dominant allele always is expressed when present. The recessive allele is only expressed when the dominant allele is not present. Use your observations from this activity to describe how codominant inheritance patterns differ from dominant recessive inheritance patterns. So you're trying to figure out what we were studying with Mouse Genetics 1, where they had black fur or white fur, compared to this. And so you're trying to say, okay, if in the mouse genetics activity, if they had black fur and black was passed on, black always showed up. But in this case, if they have red feathers or white feathers, what's the difference between that? What is being passed on to that next generation? That's what it's asking. In activity B, all right, so I'm ready to start activity B. Activity B is codominant crosses, so trying to develop some sort of predictor as to what the offspring will look like. To get the gizmo ready, we're supposed to click clear. We're supposed to drag the remaining chickens from the holding cages into the parent boxes. So that's 1 and 2. So probability. Probability is the likelihood that a specific event will occur. Scientists use probability to predict the outcomes of different genetic crosses. So the question you're faced with here is, how can you use probability to predict the outcome of a codominant cross? And number 1 tells you to do this, do a model. So a Punnett square is used to model the possibilities of offspring's genotypes and genetic crosses. The parent genotypes are written on the top and on the side of the square as shown on the right there. The possible offspring's genotypes will be filled into the boxes as they started to do in the top left. The first square is filled in for you, and they want you to fill in the remaining squares. Note, FR/FW is the same as FW/FR. So number 2 says analyze. I'll give you a second to do that there. Give you a second to do number 1. Number 2 says analyze. A homozygous chicken will have the same alleles for feather color, and a heterozygous chicken will have 2 different alleles for feather color. Are the parents homozygous or heterozygous? And then explain how you know. So if you look at the 2 parents, that means the 2 genotypes that you put on the outside of the Punnett square. Do they carry the same traits, or are they 2 different traits? That's what you're putting down for 2A. And then for 2B, it says, what are the possible genotypes for the offspring? So what you'll do is you'll look at the inside of the Punnett square, and you'll kind of figure out what are the possible combinations. So you might have more than 1 there. And then letter C says, will the offspring be homozygous or heterozygous? And then you might have other possibilities for that as well. All right, as we go down to number 3, now we're going to be doing some calculations. Now the calculations aren't going to be hard. They'll be pretty easy. Mostly factors of 4, so 1/4, 2/4, stuff like that. So I'll read this to you. Punnett squares can be used to predict probable outcomes of genetic crosses. To calculate probability, divide the number of one kind of possible outcome by the total number of possible outcomes. For example, if you toss a coin, the chance that it will land on heads is 1 out of 2. The probability can be expressed as 1/2, 0.5, or 50%. So look at your Punnett square above. How many total possible outcomes are there? So total possible outcomes, not genetic possibilities, but there are 4 squares, so there are 4 possibilities. So it would be how many out of 4, in other words. So if you go down to B, you're saying, all right, FR/FR would be how many out of 4. FW/FW would be how many out of 4. And then FR FW would be how many out of 4. And if you're confused on this, you can always send me a message and I will try and show you what you're looking at there. But in other words, you should just be putting down the numbers that you see up in the top of that Punnett square that you've done at the top of this page. C, as I look at C now, 3C, what is the probability for each of the following outcomes? So now you're putting in either the fraction, the decimal, or the percentage, as it was kind of saying up there at the top when it was given your instructions in number 3. So if it was 1 out of 4, that would be 25%. If it was 2 out of 4, it'd be 50, and 3 out of 4, 75, 4 out of 400. All right, so activity B extended. This is going to be pretty much all you got to do is about 4 more problems. So number 4 says use the gizmo to test your predicted outcome. So now you're trying to figure out what is going to happen when you breed 2 basically red and white chickens. So it says turn on statistics. So we'll turn on statistics now. We've already done that. And show the approximate percentage. Click breed, and then what are the results of the cross? So I breed them, and it says I would have 2 red, 2 white, and 1 red white. And then that says 40%, 40%, and 20%. Now, if I was to do this multiple times like we did in the mouse genetics one, those percentages might change, but the probability is probably going to be pretty close to what we found in our Punnett square above. Number 5, evaluate. Did your results of the cross match your prediction? If not, why do you think that was the case? And so that's where you go back to what you had on the previous page with your Punnett square, and you figure that out. Were you close to your prediction or were you not? And then finally it says collect some data. Number 6, click breed 19 more times. So we're going to do this 20 times until you have generated 100 offspring. So down where it says total, I'm going to get that to where it says 100. And now I will look at my percentages, and my percentages should be a lot more similar to my original prediction, right? What should have been 25, 25, and probably about 50. And then finally, 7 says compare. It says breed until you get at least 1,000 offspring. Compare the statistics to the gizmo with your original prediction and how close were they. So we'll do this as fast as we can. All right. Were we close? Yes, no, maybe so. And then draw a conclusion. Each time you bred the parent chickens, you completed a trial. A trial is a single time that you've conducted the experiment. Random chances often cause identical trials to have different outcomes. Because of this, scientists repeat experiments many times in order to make sure that chance alone is not responsible for your results of your trial. So we're not just saying that in trial 1 you notice that we didn't quite get the results that we thought we were, so after multiple trials our results kind of matched what we predicted. Hopefully this helps you out a little bit. If you have any more questions, send me a message.